Hi, my name is Maryna Chepeleva. I'm just finished my master in Belarusian State University. This work is collaboration with Luxembourg Institute of Health, and the topic is simultaneous deconvolution of bulk sample and single cell data leads to improved characterization of tumor. Examining a tumor in tissue, we see that the tumor cells are located among normal cells mixed with immune cells, fibroblasts, and others. Studying such data, we are dealing with high heterogeneous data. There are three levels of heterogeneity: native sample variability, tumor heterogeneity due to high mutation rate, and technical heterogeneity because of complexity of experiments. One of nice methods for heterogeneous data is independent component analysis. It was developed for solving cocktail party listening task. When we have several independent sources of sound, we can record them on several microphones and get mixtures of signals. ACA found a robot decomposition of input mixed signals and returns as independent signals as possible. ACA needs a lot of data, but the advantages are correction of technical biases and batch effect, and the ability to capture biologically relevant signals. In this work, we use the cancer genome atlas data of glioblastoma and pancreatic adenocarcinoma. We had gene expression matrix, and independent component analysis decomposes this matrix into two. First, in genes component dimension. Here, each column or each component represents mutagene. We use enrichment analysis to find corresponding biological processes. Second, weight matrix contains component coefficients for each sample. It can be used for prediction models. The improvement of ACA is developed parallel consensus independent component analysis algorithm. The idea is multiple decomposing of expression matrix E and averaging of the coefficients of corresponding components. This will improve the stability of algorithm. For patient classification, we compared random forest and XGBoost models. Finally, we chose random forest due to better results. We carried out multi-class patient classification of cancer subtypes. There is average balanced accuracy, independence of number of exact components. The horizontal lines are accuracy of model trained on significant genes. As you see, the ACA-based models were better on both datasets. For survival prediction, we calculated individual risk score for each component. We used only those components that made a significant contribution in survival prediction, and then Cox regression were built. Here you can see the survival curves and the estimation of quality of survival analysis, independence of number of components. And we estimated the optimal numbers. Another approach to heterogeneous data is single-cell data analysis. Cells are sorted and then each cell is sequenced separately. This makes it possible to observe the variation in subpopulations of cells. In bulk sample experiments, this is impossible. In integration analysis, we had glioblastoma single-cell data and normal pancreas single-cell data. The idea was decomposing expression matrices and then matching S matrices obtained on bulk cancer samples and single-cell data. There we found highly correlated components. That means that we can take our important single-cell metadata as a cell types and then share it to bulk samples component and use it to interpret prediction models. Comparing single-cell and bulk samples of glioblastoma, we found some high correlated components. Two of them were related to cell cycle biological process. Analyzing pancreas normal cells and adenocarcinoma bulk samples, we put in line single-cell components with cell types. Here you can see the example of diagram. And some of these individual cell types signals were found in bulk samples. And also we found high correlated component related to technical effects. Then we estimated required number of components to extract important signals. Pancreas individual cell type signals, and all these signals were detected in bulk adenocarcinoma samples decomposed on 20 components. Then required number of components to extract cell cycle component, and extract component is related to technical effects. The main results: we get the optimal number of components for cancer subtype classification and prediction of survival. We've shown that deconvolution with ACA allows to sharing annotation between bulk and single-cell data. To the most correlated components from tumor single-cell data represented cell cycle. We extracted 4 individual cell subpopulation signals in bulk samples, and we estimated the minimal number of components for extracting meaningful signals. I want to thank all the people who participated in this project. And thank you for your attention.